In this video, we will be talking about two main things. One is our biotechnology topic on designer babies. The second is how designer babies relates to a lab we did in biology class known as the P-Glow lab. First off, we will talk about how designer babies can create a world that is free of genetic disorders. Sadly, not everyone can afford designer babies, which is why our world cannot exactly be this way. Furthermore, the designer baby method cannot necessarily create the perfect baby, but it can make a baby practically free of genetic disorders. A designer baby can be synthesized through two types of in vitro fertilization, known as IVFs. In a typical in vitro fertilization, a sperm fertilizes an egg outside the female's body, so it can be worked on in the lab. In the lab, scientists can analyze chromosomes to determine the gender and possibly find certain genetic disorders. To form designer babies, the unfertilized eggs and sperm are fertilized into candidate embryos. Each embryo's karyotype and genetic sequence are analyzed before isolating an embryo that is disorder-free. Alternatively, the sperm's genotype can be analyzed for disorders to confirm the formation of a healthy embryo. In the future, however, doctors may be able to eradicate genetic disorders by replacing faulty DNA in a sperm with ones from the human genome. This method, known as gene therapy, is currently only applicable to animals. Therefore, designer babies are still primarily formed using in vitro fertilization. Next, we will be talking about how designer babies are very much related to a lab done in biology class known as the P-Glow lab. First, we will go over how this type of bacteria transformation lab actually works. We start off by making two different solutions in test tubes, one known as plus DNA and another minus DNA. In the plus DNA, transformation solution, bacteria, luria broth, and plasmid are added. In the minus DNA tube, only transformation solution, bacteria, and luria broth are added. The plasmid in the plus DNA tube will allow those bacteria to be ampicillin resistant and contain the gene for green fluorescent protein, known as GFP, to let the bacteria glow in the presence of arabinose sugar. Once the plus DNA and minus DNA tubes are assembled, 4 petri dishes are made. One dish has only agar, 2 dishes have ampicillin and agar, and the last dish has ampicillin, agar, and arabinose. The minus DNA solution is poured on the agar plate and the agar and ampicillin plate. The plus DNA solution is poured on the other 2 plates, which are ampicillin and arabinose plus just one with ampicillin on it. After waiting for approximately 24 hours, we see that the minus DNA and ampicillin plates have no bacteria on it. The minus DNA plain plate, however, has a layer of bacteria, and the plus DNA plate with ampicillin has small dots of bacterial colonies. And finally, the plus DNA plate with ampicillin and arabinose sugar have small dots of bacterial colonies that glow green. The plus DNA plates all had bacteria because they contained the plasmid that was resistant to the ampicillin. One minus DNA plate had no bacteria because ampicillin had killed them, and the last minus DNA plate had bacteria because no factors had to change it. One plus DNA plate glowed green due to the arabinose sugar. Now that you know what occurred in the bacterial transformation lab, you might be wondering now, how does designer babies actually relate to the P-Glow lab? In this lab, we used a genetically modified plasmid with chosen specific genes on it, which would allow those bacteria to survive better. In designer babies, the doctor must choose the right sperm to create the baby that will not have any genetic disorders. So, the sperm should have not been affected by non-disjunction. Hence, because both the plasmid and the sperm aid in the host's survival, designer babies are very similar to the P-Glow lab.